Think Forward. Think Research Channel.
So now it gives me a particular pleasure to introduce our distinguished speaker today. It's very appropriate for speaking at our center's premier lecture activity because he lives at the interface between health, between academics and industry. Our speaker is Dr. Bruce German, who received his PhD in his early postdoctoral training at Cornell and joined the faculty at the University of California in Davis in 1988. He is a professor and the inaugural chair of the John E. Kinsella Endowed Chair in Food and Nutrition and Health at UC Davis. And his research interests are very broad but integrated with regard to the function of dietary lipids, the role of milk components in food and in health, and the application of metabolic assessment in personalizing diet and health. So it's my great pleasure to introduce Dr. German. Please.
Thanks very much for that wonderful introduction. It's actually delightful to be back here in Kentucky. Ironically, many of the ideas I'll be sharing with you came from discussions that I had over the years with Gazer Bruckner. So if you like what you hear, then it's him. If you don't like what you hear, it's still him. I also have to acknowledge some associations. I'm at the University of California, Davis, but I'm also an advisor at Nestle, at Tethys Diagnostic Company, director of the Milk Genome Consortium, and on the boards of the Food Materials Network in Canada and Physic Ventures. So we're clear. And this is where you get to spend time if you're advising Nestle. It's a mountain in Switzerland. So I want to give you some trends in biology, how basically the science is moving forward with respect to nutrition, diet, and health. And basically there are three trends that are clear that we can now say are underpinning much of life science. One is prevention. This idea that we can now begin seriously scientifically to recognize predispositions of individuals and begin to act early enough that we can say we're genuinely preventing the development of diseases. But in tandem with that is the recognition that we're going to have to individualize, that the solutions will be required at a much more personal or individual level. And interesting, one of the aspects of science and life domain is biology itself is becoming an important tool set, and I'll give you examples. Just to basically summarize, the 20th century from a scientific and an industrialization perspective was genuinely the success of chemistry. From John Dalton's original atomic theory, the great success of being able to recognize molecules, synthesize molecules was the great success of science of the 20th century, and its industrialization as chemicals, from herbicides, pesticides, and fertilizers to antibiotics and therapeutic drugs. Chemistry underpinned the success of life. And in fact, if you look around and under you, the success of synthetic chemistry has genuinely changed the human condition. And we have a lot to be thankful for in a nutritional perspective because of the success of this era. The 21st century, however, will begin to recruit a new area of science, and that's biology. Of course, Charles Darwin, its patron saint, the evolutionary process, and genomes will become scientifically the knowledge base on which we'll do that. And we will begin to industrialize them. In fact, one of the first examples of this is we will literally use living organisms as industrial carriers of value. The food industry is already selling live organisms for their value. Yogurt, sequenced genome intact microorganisms that give rise to both a nutritional value and an organoleptic value is the first indication that the industrialization of biology is already happening. And these have many implications to nutrition. What we are understanding with biology is the variation in the human condition. Nutrigenomics is beginning literally to give molecular detail, genotype detail to the variation in response to humans to different diets. We are changing the way we view nutrition. We no longer think of nutrients solely as building blocks and cofactors. Because of genomics and the ability to look at the expression of genes, we are beginning to describe food and nutrients for their ability literally to affect the expression of genes. We're using evolution as a principle to begin to understand the emergence of humans, our requirements for vitamins, but also the emergence of a variety of foodstuffs, and I'll talk about milks. And another interesting aspect of evolution, the evolution of the bacteria in and on us, and how intimate they are as partners in our health. We'll also talk about that. So this new biological perspective is moving very much into nutrition. One of the major consequences, one of the great benefits of the 20th century, the industrialization of chemistry, the understanding of molecules as essential nutrients, was a spectacular change in life itself. By every historical criterion, most modern humans will live 3 lifetimes. Typically, humans lived, on average, 25 years. That was life. Today, most humans will live 3 intact lifetimes because of the drugs available, the various sanitary, hygienic conditions made possible by chemistry. That's the good news. The bad news is that you are promised 3 lifetimes. You aren't promised 3 healthy lifetimes. The health of those second and third lifetimes will be very dependent on how well you nourish and, in essence, guide your lifestyle. So the healthy part of your 3 lifetimes is literally up to you. So just to give us a sense of how we got where we are today, the 20th century and the literally disassembly of food into discrete molecules, nutrition became a molecular science. And that was a spectacular enabling decision because it allowed nutrition as a field to literally use what engineers call a fault model. You could feed animals all molecules and take one away. If it's necessary to function, then the animal system fails. Using that simple fault model, all of the essential nutrients were discovered. The vitamins, the minerals, amino acids, and fatty acids, a spectacular achievement of science. We know all the essential nutrients. Keep in mind, we're still debating how many planets there are in the solar system. So on balance, this was a great success. But in getting it, there were certain assumptions made that still persist in our thinking today. First, the research was reductionist, 1 molecule at a time. That was necessary to its success, but we still have that reductionist philosophy. The application was to the entire population. The good news is the same recommendation for essential nutrients works for everyone. And in fact, everyone has the same essential nutrients. It's not like some of us can make vitamin C and some of us can't. So it was a population-wide decision. And the implementation could be very molecular as well. You could have a bad diet all day, and from an essential nutrient perspective, you could solve it completely with a supplement or fortification at the end of the day. Those key success features of essential nutrients are still persisting in our thinking today. There were consequences. Without fear of deficiency, because the industrialization of this knowledge brought it to the food industry, and in fact not education, we didn't decide to teach people where iodine was, we iodized salt. What that did was it converted nutrition to an elective. That is, students could choose to take nutrition and health or not. And as an educator, I can tell you what students do to an elective. They elect not to take it. As a result, we are clearly profoundly ignorant about diet and health. All you have to do is tune into TV late night in California, and the things that they promise to expand, contract, alter about your body clearly reflects the population does not understand what they are being victims of. And finally, a very interesting aspect is nutrition and food as scientists separated. They literally divorced. Nutrition, a molecular science about nutrients. Food became much more occupied with the organoleptical properties, the safety properties, and the production values. And they separated. So food and health became separate fields. And that was a mistake. Another consequence, because you didn't have to worry about essential nutrients, they were in a variety of foods, the food marketplace, one of the most dynamic markets in the human condition, began to evolve on purely a hedonistic basis. What I mean by that is pure delight. The marketplace competed, every food competed for the consumer's delicious. And foods became more and more delicious. And that was the driving force for competition. Foods that weren't delicious couldn't compete. What we get is a very delicious food supply, but is it healthy? And we got choice. This is an image of a typical supermarket in the United States illustrating just how much choice. Now remember, this choice is available to consumers who have no education for the most part and are completely ignorant of the implications of their choice to their long-term health, and they're living 3 lifetimes. So this set up a very bad problem. In fact, when you look at health and the investment we've made in it, the resources we've brought to bear, the knowledge we have, we should be the healthiest humans in the history of the world. We should be enjoying the fruits of all that knowledge. And some are. But unfortunately, increasingly large numbers of the population are suffering from diseases that are literally the result of bad choices for diet. We should be much healthier and we're not, and food's the problem. So how are we going to move forward? So we have a wonderful challenge. Change food, change diets, prevent disease, not wait for it to happen. It sounds very attractive. You could run for president on a platform like this, and they are, but it's much more difficult than you'd think, because prevention is very complicated. First of all, health isn't easy. Remember, if you're talking about disease, you've got something catastrophically wrong, you need to find it and fix it. But if you're looking to prevent disease, people are already healthy. You're looking to make them healthier. That means you're trying to improve the risk of all diseases. If I lower the risk of one thing with a food or ingredient, and in so doing, I increase the risk of anything else, even in a subset of the population, I haven't made you healthier at all. That's the first real challenge to prevention. You've got to get it all right. That means you have to be able to measure health and its consequences across all aspects of health. Second, we're not the same. Now, normally when you see images describing people who are different, you see people who are healthy and people who are ill, and that's the difference. But in this case, images from that very prestigious scientific journal, Sports Illustrated, these are the elite athletes from a wide variety of sports. Literally, they're the healthiest among us. And you can't look at these images without agreeing. We're different. It's a biological reality. But there's also a philosophical one. Imagine that you could make, in fact, a Purina human chow that would make everybody healthier. It would make them more the same. But that's not what we want. The differences between us in our lifestyles, in our structures, we cherish those differences. We enjoy the diversity that life affords. And so if we're genuinely going to make people healthier, we have to make them healthy the way they define it, not the way we impose it upon them. So philosophically, we have to get the science so right that people can enjoy the health that they want for themselves. And that's the challenge. And third, translation. It takes decades for science to move from basic research, discovery, to the bench, to the bedside, and the other direction. As we are now understanding, translation is a very long process. Typically, by NIH criteria, 30 to 50 years That means that if you young students make a discovery, guess what? You will not personally take advantage of it. That's a real problem. So how can we solve these three problems? I'm going to give you our philosophy, the way we pursue it at Davis, and as a model for how we think we can move forward. So, one obvious question, how diverse are we? What are the variations in humans? What's our potential? Not just what are we stuck with? What are the diversity of environments? How different would we like to be? And finally, diversity of food. We are stopping to think about food as the 20th century. What is it that's in food? But the 21st century. What can foods actually do? A very different view. So, what are we? Phenotypically, we are, in fact, the combination of genotype, what we got from our parents, and our environment. That interaction is what we understand is the basis of life, the equation of life. But nutritionally, we are also learning that you interact with your environment through your life. Your environment affects the expression of your genome, and it has persistent effects. So actually, phenotypically, we are the combination of our genotype, our environment, and the history of that interaction throughout literally our entire lives. That's what phenotype is. So we have to be able to capture that level of diversity. These are actually perfect phenotypes. These are my daughters. Now, in many respects, they are the product of genetics. They'd like to be tall, as all young women would today, and they're not. And they know why. It's the father. Actually, if you want to know your faults, have a daughter. She'll tell you every single one of them. But it's not just genetics. It's also the environment, what they choose to do, how they choose to eat. In fact, same genetics, 1 and 21. It takes about 20 years, 10 million calories to make a perfect daughter. Same genetics at 1 and 21, clearly not the same phenotype. Should they eat the same? No. So we have to understand certainly genetics as one of the ingredients of phenotype, but we have to be much more explicit in the other variables. We have to understand phenotype itself. So what we need to do is measure it. We need accurate assessment of health itself as phenotype, and we don't want simply diagnostics of disease. For my daughters, it's too late to wait until they have the disease. I want to know long before it happens. That's clearly the goal. How will we do that? Well, remember, we have to assess phenotype health in this comprehensive way. I can't look at one thing, one biomarker of disease. I can't, in fact, build a health assessment program on diagnostics of disease. It just isn't good enough. Measuring everything sounds literally beyond the dreams of science, but in fact, it's mainstream science today. That's exactly what the genomics revolution has done. It's provided the means to get comprehension and molecular detail simultaneously. That's what genomics is, that's what gene expression does, that's what proteomics, metabolomics, that's the science of today. So the possibility, at least conceptually, to measure everything about health is available. But what we think is most important is to measure closest to phenotype, and what we approach is metabolism. Looking at metabolism with sufficient accuracy and comprehensive perspective that we can look at all aspects and measure it explicitly. There is a human metabolome initiative going on around the world where all of the metabolites within humans and their various biological fluids are being assembled and annotated. So it's a worldwide operation and it's ongoing. What we do at Davis is simply lipids. We're lipid geeks, I apologize, but our perspective is to understand lipid metabolism. We can build the tools to measure just lipids, and as our role in this overall global enterprise, get the lipid compartment right. So what we do is take biofluids, separate them into various lipids and measure them very exactly, and then attempt to build the biology representing the variations in those metabolic profiles. One thing is you have to realize that if you're assessing health, accuracy is critical. Diagnostics means you can ignore the average variation in the population. You're looking for those rare disease-based excursions. But for health assessment, no one's diseased. You have to be able to capture the variation within the population. Here's an example of just that. I've taken one metabolite we measure, arachidonic acid, in a particular phospholipid class in blood, and these are measurements of humans as part of a clinical trial on 2 separate occasions. What you can see is the level of these specific metabolites are quite constant within individuals, but vary between individuals. Our metabolism is part of our signature phenotype. It's measurable, but in order to capture the variation between people, you have to be very accurate. But this illustrates it is possible, but you have to build platforms that are capable of measuring the molecules accurately in an absolute sense. When you do that, you can begin to interpret those variations and annotate why are people high and low. So our model is this. We would like to build the technologies to be able to measure metabolites, build databases using those measurements across a large number of phenotypes, and then understand the variation in those metabolites in terms of health and outcome. Now, it sounds like an awful lot, but it's all been done before. For cholesterol, the technologies to measure cholesterol were established, and then those tools were brought to measure the cholesterol levels of hundreds of thousands of people, and it was recognized that when your cholesterol level is high in blood, this predicts a metabolic condition that predisposes people to heart disease. And if your cholesterol is low, you have less risk. And then the biology of understanding how cholesterol was regulated, the genes, the pathways, etc., were all assembled. So we know what to do, we just have to do it again, but we have to do it for a larger number of metabolites. But the number is not infinite. It's about 5,000 total. Keep in mind there's over 5,000 institutions of higher learning in the United States. So in fact, it is not an unmanageable approach. And the good news is it's the same goal for everyone. So for the pharmaceutical industry, they have to look at this because they're building lifestyle drugs, and they need to understand both efficacy and side effects across the board. Ask Vioxx. Vioxx worked. It had side effects. So the pharmaceutical industry is desperate for these same approaches. Nutrition and food. You need to be able to understand what are the consequences of a food in all aspects. Nutrition and food are looking to build these same databases. And finally, lifestyle and exposure. The environmental community needs to know what are the long-term consequences of a particular environmental exposure. And even the lifestyle industry. What different exercises should people actually pursue? So everyone in life science, in the business side and the science side, is going after the same thing. Understand and measure health. So we look at lipids, and we look specifically at blood as a bioreservoir, and all of the different tissues that contribute and remove lipids are then being sampled in a very surrogate sense by our measurements. I'll give you a couple of examples of how we've used these technologies to build an understanding of metabolism, of lipids, and how that can be brought to practice. So, give you 3 examples. One, phenotype discovery. We're trying to discover the basis of a phenotype, NASH, the development of fatty liver and its progression to steatohepatitis. Diversity discover, we're trying to look what are the variations in people that could be basically the reasons why they respond differently to diet, as a means to begin to say what are the metabolic reasons why people in fact differ. And finally, a target discovery example where we've actually revealed a signaling pathway that was not recognized without these techniques. So, in terms of fatty liver, this is an increasingly problematic aspect of human health. The growing incidence of obesity seems to be being followed by a growing obesity of livers. In some people, this halts at fatty liver, but in some it progresses to steatohepatitis, and in some actually to cirrhosis and the loss of the liver. We need to understand what is it metabolically that's going wrong, and can we begin to intercede to prevent it long before it actually reaches the end stage. So we've been using an animal model for this in collaboration with Jackson Labs. They have a murine model, a mouse for diabesity. Overweight, fat, diabetic mice. It was developed as a discovery tool, and this particular model illustrates both genetic predisposition, metabolic dysregulation, and a nutritional rescue. So this is basically clinical biomarkers of 2 drug classes on this particular model. And this is cholesterol, glucose, and triglycerides, the hallmark biomarkers of what you would like a drug to do in an overweight pre-diabetic model. And it looks like both these drugs are doing exactly the same thing and working both very, very successfully. Unfortunately, in this model, a PPAR gamma agonist is having a rather discouraging effect. It's causing fatty liver. And in fact, these animals are dying from the consequences. Beta-adrenergic receptor agonist is not. So a very different phenotype, and we wanted to know what is the basis of this metabolic dysregulation. So first thing we did was measure the lipids in the various compartments of these animals using these broad comprehensive tools. So this is a visualization of the sorts of data we get. Every single cell is a different fatty acid in a different class of lipid across various tissues. If the cell is black, that means there's no statistical difference between the two animals, the drug treated and not. If it's green, that means it's statistically lower as a metabolite in that fluid, in that animal, in that class. And if it's red, that means it's elevated. Now, what you can see, for example, is triglycerides in liver are way up, and triglycerides in the blood are way down. This is what makes the biomarker that it looks like a successful drug, but here's where you see the accumulation of triglycerides. But look at all of these fatty acids and all these differences. This drug is having a major effect on metabolism in these animals. But how do we figure out what are these differences related to that could cause the problem? So we began to look with a variety of collaborators at the basic biology of lipid synthesis and secretion from the liver. We're working with Steve Zeisel on an interesting knockout model of a phosphatidylcholine synthetic pathway. You make phosphatidylcholine in the liver by two pathways, one from diacylglycerol, which is choline dependent, and one by the methylation of phosphatidylethanolamin through this pathway. This is a knockout model, so this pathway doesn't work, but phosphatidylcholine normally proceeds easily through this pathway. And this pathway is necessary to getting fat out of the liver. If you can't surround a lipoprotein with phosphatidylcholine, it can't get out. And interesting, looking at the data, you can see this, when this is knocked out, is a choline-sensitive pathway. When the animal is getting lots of choline, this goes fine, and there's no phenotype. It's all black. It's the same no matter what. However, as you take choline out of the diet, now the liver begins to accumulate the fat and the plasma loses it. So we're beginning to understand exactly how this works. Now, it turns out there are diagnostics of this metabolism. This pathway assembles different fatty acids on the phosphatidylcholine than this pathway. So in plasma, we can measure the phosphatidylcholine and know quantitatively how much these pathways are working and which one is working successfully or not. So using this model, we now have the diagnostics of the entire process. And in fact, we've brought this forward as a model to look at humans. So we can now look at the metabolic precondition of the successful production and secretion of lipid from the liver, and we can diagnose in those individuals when these pathways aren't working even before they begin to accumulate fat. So we can diagnose metabolism literally by measuring composition in blood, exactly what we set out to do. And we've recognized that if this is a problem, we can even use a nutritional rescue to push the pathway and get it to be successful. So taking that to a metabolic perspective in these mice, we've got fatty acid synthesis and triglyceride up, red, but they don't get out. It's down here. In fact, the adipose are accumulating it, and that's why the mice are actually gaining weight. So we now understand what the drug is doing is inhibiting phosphatidylethanolamin methyltransferase. This particular model has an unusual susceptibility genetically to this step and to this drug. So we have this genetic susceptibility to the drug revealed phenotypically, but we can diagnose it in real time, and we can do it before any of the long-term deleterious consequences have happened. So our goal of understanding metabolism, building it into a diagnostic, is realized in this condition. In fact, we've used a variety of other lipids to basically map the entire process. Very early, the fatty acid metabolism changes are the only thing that are going wrong. but you can detect it. As it moves through fatty liver, we begin to see inflammatory lipids appear, and as it moves to NASH, we see a variety of lipids that are now signature. So we can tell where people are in the process and, in fact, begin to develop strategies at each step. So understanding metabolism, measuring it, allows you to basically phenotype people for this condition. And the good news is we only use up 5 metabolites to be able to fully describe these various pathways. So in terms of this idea of discovery of differences between people, we've actually been taking a somewhat different approach to nutrition, measuring people in the fed state and feeding them different fatty acids and following their metabolism. And one of the things that we were unprepared for is how differently people respond to omega-3 fatty acids. I think you know nutritionally, it's on the news everywhere, people should eat more omega-3s. Well, in fact, people respond quite differently. This individual is a high omega-3 status coming in, actually is a fish consumer, and when given 18-3, the precursor in the diet, successfully converted to product. This person, very poor omega-3 status to begin with, but nonetheless is able to convert. This person doesn't have omega-3s because this person clearly doesn't eat any. This person doesn't convert at all. 3 healthy people, very different reactions to diet. We need now to understand the genetic metabolic basis of this. So we've discovered a diversity in humans that was not recognized before. And then finally, new pathways. Working in collaboration with Gokhan at Harvard, he was looking at the communication between adipose, liver, and muscle. And it turns out that a particular fatty acid, palmitoleic acid, a product of lipid synthesis in adipose, is actually signaling to the liver. And he's dubbed these lipokines. This discovery was only possible because you could measure very accurately all of the variations, and this particular fatty acid literally spoke volumes by its unique abundance. So again, from phenotype discovery, diversity discovery, and now pathway discovery, measuring metabolism is entirely possible, and once you have the platforms in place, you can measure it and build diagnostics with it. So we're going to be personalizing health. Everybody, when I talk to them about that, they say, that's never going to happen. No one is going to personalize health. There's just too much technology, too many hurdles. Well, let me give you an example of personalizing available today. I can go to golf course, California, and I get one of these motorized carts. And there's a very personalized system in it. There's GPS in the golf cart. Think about that. That means there are satellites orbiting in space using atomic clocks, Einsteinian mathematics, to communicate with one of the largest databases in the world, a map of the world. All of this delivering a very personal technology, the GPS in the golf cart. But what's most astonishing about that is the value proposition. What do I get for all that? For the space toys, for everything else. What am I able to do? How is the human condition so enriched by all this technology? What you get is, when you're standing beside the cart, you know how far you are from the ball to the green. That's it. That's the whole value. So, I would ask the overweight, diabetic, impotent, hypertensive, atherosclerotic golfers, what would they prefer? Clearly it's possible to personalize health. We haven't yet. If we can personalize GPS in golf carts, we can certainly personalize health. So, what we will do as we do it, and in fact many of the large companies around the world are beginning to realize there will be value created. Measuring human health will become an industry. In fact, General Electric, Philips, Siemens, Roche, Agilent are all moving aggressively in this direction to go from diagnostics of disease to assessment of human health. The consumer of one. This is what they realize is their future. I'll give you some examples. Actually, GE and Nestle are working together on GE's IDEX technology to measure originally bones. So you can measure and get a whole pattern of your bone, and this was originally developed as an osteoporosis measurement. In fact, they began to see body compositional artifacts, and they realized they could actually get a map of adipose and muscle using the same technology. And it's available. And actually, I was playing ice hockey last year, ran into a particularly unpleasant opponent and broke 2 ribs and partially collapsed my lung. And I thought, well, this may be bad news. I may be actually getting osteoporosis. I didn't think the collision was that bad, but clearly it was a problem. But went and had my bones measured in the GE machine at Nestle, and good news, actually I have very high density of bones. Bad news, of course, my wife was right. I broke my ribs out of sheer stupidity. I shouldn't be playing ice hockey against 25-year-olds. But this is technology available today being used to measure health. Literally the distribution of muscle and adipose and bone in normal human beings, and developing foods based on this. It's also possible to take vast amounts of data and reconstruct them, visualize them as interpretable images. This, in fact, is not an image. This is a series of NMR spectra captured and then reconstituted mathematically into an image of the heart, in this case gated by the beating of the heart so you can see it in function. And in fact, this is my heart, perfectly non-invasive, and apart from the small scar there where Susan Scott broke my heart in 8th grade, it's normal. I can send this image anywhere and cardiologists can interpret it. So in fact, this notion of measuring using very high density information is possible today. We just have to take advantage of it. In fact, we have taken the technologies in the lab, spun a company out of the lab, Lipomics, and they're now working with Tethys to make diagnostics of the risk of developing diabetes. So you can take these technologies very quickly into the marketplace to just give people enough information to act upon their current phenotype. So diagnostics of health and predisposition is possible today. We can actually do this. So assessment, understanding the variation in health and predicting trajectories is increasingly possible. Unfortunately, that's the good news. That's the easy part. What we would like to do now is intervene. We would like to build diets that literally make healthy people healthier. The problem is we do not have a strategic model of that. For a disease, we do, a wonderful one. Diseases are defined by targets, the catastrophic failure. You use the target to develop diagnostics to identify those people who have it. You use the targets to have high throughput diagnostics and high throughput targets for therapeutics, for small molecule discovery of drugs, and an entire pharmaceutical industry, highly regulated, and clinicians to deliver drugs. For disease, we have a strategy, and it works very successfully. But for health, we have healthy people, we'd like to make them healthier. What are the targets? How are we going to use ingredients in foods that actually make people healthier? We have no strategy. So we can ask how we might go forward. And of course, it's the 21st century. We have genomics, we have genomes of humans, plants, animals, microorganisms. These are the knowledge repositories of evolution. We can begin to ask the questions, what in fact would likely be nourishing from the evolutionary process. If you do that, there's some obvious examples that are being used today. Plants are being used by the pharmaceutical industry. And the best example of that, Merck bought the genetic rights to Costa Rica. They own the jungle. Right? Why? The probability of the jungle containing highly selective and active molecules is very high. But for drugs, typically what you want is selective toxins. Take cancer. You want a molecule that's toxic, but so selectively toxic it kills the cancer cell, leaves all the other cells around it alone, and oh, by the way, they're all your cells. Selective toxicity is what you want in drugs. Why would you look in the jungle? That's exactly what the evolutionary process has done. The constant Darwinian selective pressure on plants has been to avoid being eaten. They can't run away. They have secondary metabolism, and that's what they use. So if you're looking for drugs, plants are an excellent place to look. The chances are very high you'll find them. However, if you're looking to nourish healthy people, to make them more protected, more developmentally mature, more able to deal with the problems of environmental stressors, plants aren't necessarily the place to look. What could we use? there is something. Milk. Lactation, the process of emergence of this unusual nutritional strategy of nourishing young infants with the mammalian secretion of the mammary gland. Now, this is a model. In fact, think about it. You've got a Darwinian engine for nutrition of your dreams. The mother is literally dissolving her tissues to make milk. Everything in milk, including the water, costs her. If it doesn't provide a value to the infant, it'll be lost to evolution. But if in any way it improves the success of this offspring, and it's healthy, it's going to be retained. And this engine working millennium after millennium, year after year, generation after generation, has brought wonderful information. We just have to tap into it. So, taking this approach, we have developed an international consortium looking from scientists of a variety of places. What does the mammalian genome tell us? What isn't not how lactation works, why lactation works. What is it that gave rise to all these components? How do they work? How do they provide value? I'll give you a couple of examples. Lactation strategies are fascinating. The black bear, actually up north of you in Canada, this bear goes into the cave pregnant, goes to sleep, and then, without so much as waking up, much less going out to feed, this mother gives birth to 2 to 3 cubs and lactates and feeds them completely for 4 months. Literally in the spring, the family walks out of the cave intact. An absolutely astonishing example of nourishing efficiency. What mother wouldn't like to go to sleep and wake up with the family? The exact opposite end of the spectrum, the hooded seal. This is a seal that's born in the North Atlantic on ice floes about the size of this room, right in the middle of polar bear country, a very unsafe environment. And so the strategy is to get the pup off the ice floe. So lactation is very short. In fact, it only lasts 4 days total. 4 days, the pup is off into the open sea. But in each one of those 4 days, the mother seal transfers to the pup 7 kilograms of fat every day. At the end of 4 days, that pup is 50 pounds fatter. It's the most astonishing example of energy delivery in mammals. But throughout those 4 days of obvious hypercaloric status, that infant maintains absolute control of its metabolic regulation and does not suffer any deleterious consequences. If we could understand that, we could stop diabetes. The problems of a hypercaloric state are solved in this animal, if we could understand it. From the other perspective, this pup's mother loses 50 pounds of fat in 4 days. Wouldn't you like to know how to take 50 pounds off the average American in 4 days? It's in the genome. So that's our model. We've been participating in the sequencing of various genomes. We've participated in the bovine genome, which comes out in Science actually next week, and using genomics to understand lactation from a purely genomic perspective. For example, lactation. Remember, lactation itself is a very recent elaboration of evolution. All of the organs, the hearts, intestines, etc., were in earlier animals. The mammary gland literally was assembled from different components. And we can begin to ask very interesting evolutionary questions. For example, relative to other genes in the genome, are milk genes evolving fast or slow? Well, interestingly, relative to other genes, our mammalian lactation genes are evolving conspicuously slowly. In fact, it clearly is one of our great treasures genomically. We don't mess with it. So in fact, lactation is a pivotal aspect of the human genome. So milk protein genes are more conserved across mammals than other genes, and the genes expressed during lactation are more conserved. And for those of you who have had, I can only say in politically correct terms, something less than respect for us lipid people, I want you to know that the most conserved gene set across all mammals and lactation is the set of genes that make fat in milk. So genomically, lipids are very important. So my career path has been validated. One last quick illustration of how we've been using this model at Davis to develop new knowledge, targets of action, benefits, and how to translate it. So we've been looking at basically glycobiology. And milk has a very interesting property. Human milk, remember this model. Mother dissolving her tissues for the infant. If it doesn't value the infant, it'll be lost because it costs her. Imagine our surprise. Relative to protein in human breast milk, an equal amount of intact oligosaccharides. Just as much as protein. And oh, by the way, the oligosaccharides are undigestible by the mother or the infant. What could they be for? So we began to look very intensively at this. The structures themselves were not well characterized. So we recruited the best analytical chemist in the world in glycobiology, Carlito Labriglia. He's actually at Davis. He builds mass spectrometers solely for this purpose. If you can make a mass spectrometer so accurate, you can detect the exact structure of the oligosaccharides. So we've been using these platforms. We've measured the mass spectra. We know what the structures of all these molecules are. We've worked our way back through primate evolution, looking at the emergence of all of these structures through primates, actually all the way through marsupials. We're now measuring the oligosaccharides in the platypus duck. So this is actually a pre-mammalian process that elaborated into mammals, and in fact humans make the most diverse and the most abundant. But all mammals make them. We've worked with Agilent to produce a high throughput device, actually a nano LC system, that instead of using the multi-million-dollar technologies of Carlito, we can now, with the libraries he's developed, use a very simple Agilent mass spectrometer and do this in reasonable throughput, and anybody can use this around the world. But what do they do? So clearly, if the infant doesn't digest them and consume them, something else must. And so we recruited Dave Mills, who's an expert in the bacteria that colonize the human intestine, and we've been looking at the bacteria and asking the question, do bacteria eat them? Well, it turns out most don't. Most bacteria that do very well on lactose or a variety of plant sugars, very successfully, most cannot consume human milk oligosaccharides. Interestingly, one very rare bacteria does, and grows very successfully. This is the strategy. This is the genius of lactation. What the mother is literally doing, Bifidobacteria infantis, we found in the intestine of breastfed infants. The mother is literally recruiting this bacterium as a babysitter for her infant. She's recruited another life form to protect her infant. By recognizing this strategy, we now know a whole new way to nourish the health of individuals. Selectively nourish very specific bacteria. We've sequenced the genome of this organism. We've looked at the molecules that it digests. Turns out that the infantis genome is a larger genome than most bifidobacteria by about 20%. It has about 700 genes unique from other bifidobacteria. It clearly co-evolved with humans. We've been selecting this organism to protect infants and building the oligosaccharides that it uses to outcompete other bacteria. So, a very substantial and unique property of this bacterium, and in fact this coevolution we've recognized by the genomic sequence of particular proteins. So we've been able to assign the coevolutionary process of this. In fact, this is a very unusual bacteria in that most of the genes necessary to digest human milk oligosaccharides and live on this food source are in a single cassette or cluster of genes. Further evidence of this coevolution. So what we now have is the oligosaccharides and the genes, and we can now begin to manipulate those so we can change the way everyone could eat. So in fact, the elderly who have problems of microbial overgrowth and the poor immune system, senescent, and the inability to block bacteria by the pH in their stomach, we can in essence give the same potential value to the elderly. from an oligosaccharide perspective, breastfeed them, to use this strategy to protect their health. So, from a translational perspective, let me give you a sense of how we did this. So we started with milk, developed the oligosaccharide analytical capabilities with Carlito, recruited the microbiology with David, understood the bacteria necessary to do this, then worked with a company that develops human milk as a resource for particular products to give us enough material we could then move forward. Mark Underwood is the head of the neonatal clinic in UC Davis. We have a clinical trial looking at premature infants. Premature infants, for the most part, are born by cesarean section. They're put into an incubator, and in fact, they don't get breast milk. They are colonized by the bacteria in hospitals. This is not the way you'd like to start life. So in fact, we're feeding these premature infants with the oligosaccharides and the bacteria from breastfed infants. We've actually been working with DSM to find structure-identical oligosaccharides from other sources. Helen Raybould has been looking at the signaling in the intestine to see how these oligosaccharides and the bacteria stimulate. We've been working with Bart Weimer, who's developed a literally annotation of these molecules for their ability to bind and eliminate pathogens. See, Chen in chemistry is looking at how to synthesize very specific structures and decorate oligosaccharides the way we would want. And Eric Gershwin, head of rheumatology and immunology, is looking at the elderly in a model of immune senescence. This translation process is 4 years old. We have clinical trials going on in these molecules 4 years after we started. Why? Because it's human milk. We don't need to worry about safety and efficacy. Evolution took care of that. If this was an unusual plant material, can you imagine how long we would have to wait before we were sure that it was safe? This material is in human milk at the amounts we're giving it. So the efficacy and the safety are the key aspects that we can count on evolution to provide. We're also looking at the lipids. The lipids of milk are unusual in and of themselves, and it turns out that milk contains not only big fat globules, but little tiny globules, and these bind very successfully to toxins themselves. So we think that the lipid particles are providing the material to make HDL for infants and binding capacity for toxins. So we're starting the whole process again, looking at the lipids in milk and working around developing collaborations and translating. So again, milk can be used as a translational model for nutritionally effective molecules. So our model then from health, milk as a model, discover the molecules, the structures, the mechanisms by which they work, and the benefits to health. Oops. So in summary, the promise to improve health, we will need to personalize, and we will need to personalize phenotype itself. You will have to be measured. On the basis of those measurements, you have to gain actionable knowledge about your health now and what it's likely to become. That will guide your lifestyle, your diet, your exercise, etc. Personally, combining what you want out of life with what you need to achieve that. Interestingly, the goals to get that successful require innovative strategies for nutrition, and we think lactation, this evolutionary model, is appropriate, and we're finding molecules, structures, mechanisms, and benefits that are successful. Finally, these are the various people at Davis, at Metabolomics and Tethys, at Nestle, and the various people who have collaborated and funded this research. Thank you.
Think forward. Think Research Channel.